Good morning, everyone, and welcome to today's session of our 2026 Crops Winter Webinar Series. Today, our topic is herbicide resistance. My name is Samantha Marcino, and I'm the Crops Extension Specialist with Saskatchewan Agriculture based at the Yorkton Regional Office. CEUs are being offered for CCAs on this webinar. In order to get credit, you must attend the entire live event. There will be a QR code at the very end of the presentation to collect credit. If you missed the code and have attended live, please email us afterwards and we can work with you to get your credit. Before we start the formal presentation, I have a few items to share. Our Crops Extension Specialists put on this webinar series each year. We work out of 10 different offices across the province. Each office contains a wide variety of specialists that can help you with any agriculture-based questions. To connect with our specialists, please call your local regional office or the Ag Knowledge Center. There are 8 AgriArm sites located throughout the province that are dedicated to bringing Saskatchewan-specific information and demonstrations on new and existing agricultural practices. If you're interested in learning more, please follow the QR code on the screen. If you have ever heard the words on the radio, Saskatchewan is 98% completed harvest this week, then you've likely heard of the crop report. The CES team writes the crop report each week during the growing season based on data collected from the crop reporters across the province. I would like to thank all our current crop reporters and would also like to invite any new crop reporters to sign up. Please use the QR code. The 2026 Crop Diagnostic School will be held at Swift Current, Saskatchewan this year on July 29th and 30th. More details will be available soon. We still have 2 more winter crops webinars, as well as some other webinars put on by the range management specialists and the agri-environmental specialists. You can register today on our website at the Sask Ag Events calendar. Lastly, we want to make sure everyone is aware of the funding available through the Sustainable Canadian Agricultural Partnership. You can reach out to your local agricultural program specialist to find out what funding you may qualify for. Along with this information, we have additional resources for everyone under our handout section in the chat box area of the toolbar. These will be shared along with the recording after the webinar is complete. Today's presentations will be followed by a live Q&A after each individual speaker, and the main hub will be your webinar screen. The entire session will be recorded, so if you miss anything or wish to revisit any of the presentations, you will be able to. Recorded links will be sent out later this week in a follow-up email. An evaluation will pop up at the end of the webinar this morning. Please ensure that you fill out the evaluation, as these help us determine what topics you want to learn more about and help us prioritize research needs for the future. A few housekeeping notes to keep in mind during the webinar. If anyone has questions during any of the presentations, you may type them into the question box on the top right-hand side of your control panel. We will try to get to as many of your questions as possible. Your question will remain anonymous and will only be sent to me. During the presentation, you will be muted so that we can ensure there is no feedback that will interfere with the audio. Now we can get started with our first presentation. First up, we have Dr. Charles Geddes. Charles is a research scientist with Agriculture and Agri-Food Canada based out of Lethbridge, Alberta. Charles developed a strong connection to prairie agriculture from a young age, having grown up on a mixed farm near Pilot Mound, Manitoba. He graduated with a Bachelor's of Science in Agroecology and a PhD in Plant Science from the University of Manitoba. Currently, Charles leads the Weed Ecology and Cropping Systems Research Program, which focuses on discovery, characterization, monitoring, and management of herbicide-resistant weeds in Western Canada. He has contributed to the broader agriculture community in several roles, and among them, he currently serves as first vice president of the Canadian Weed Science Society and co-lead of the Prairie Weed Monitoring Network. We can start whenever you're ready, Charles.
Okay, can you just confirm you can see my screen there?
Yeah, looks good.
All right, excellent. Okay, good morning, everyone. Thanks for the wonderful introduction. And today I'm going to be talking to you about herbicide resistance, along with my 2 colleagues that will be following up, talking about some of their research on herbicide resistance as well. And so we'll jump right into it here. first of all, what is herbicide resistance? So herbicide resistance is the inherited ability of a plant to survive and reproduce following exposure to a dose of herbicide normally lethal to the wild type. And so essentially what that means is that initially the plants were susceptible to the herbicide, and they have evolved to overcome that herbicide at rates that would normally be lethal to those original plants. And so why do we see herbicide resistance showing up? And I mean the overall reason is because contemporary weed management programs lack diversity, and really we've focused predominantly on herbicides, and we've been using this easy button here going on 7 or 8 decades now, and I mean lack of diversity in the weed management practices that we use, but immense diversity in weed communities mean that weeds can take advantage of that and overcome low diversity weed management programs. But really, I mean, there are a lot of reasons why farmers would use herbicides, and I mean they're highly effective in general on susceptible weeds. They're quite cost effective, and they allow for savings in time and labor, just to name a few. Um, so what does it look like for herbicide resistance? In Canada, we're sitting in third place, you could say, for the greatest number of herbicide-resistant weed species. Over here on the right, you can see a graph that's showing the increase in herbicide-resistant weed species over time, currently sitting at 44 across the country. On the left, we're looking at unique herbicide-resistant weed biotypes. And what I mean by that is the combination of a weed species resistant to a herbicide mode of action. Currently sitting at about 87 across the country. And you can see that that's increasing rather linearly. So if we plot a trend line here, we're discovering about 2 new unique herbicide-resistant weeds every year across Canada. And so increasing herbicide resistance, of course, is important when we consider that there are a limited number of herbicide sites of action or modes of action to manage these weeds. Here we're looking at the timeline for commercialization of different sites of action at a global level. You can see denoted by their various different group numbers. And one of the things that I usually point out here is that, I mean, we've been going on 4 decades with very few new herbicide sites of action being commercialized and available for use. You can see essentially the plateau there since the 80s with 2 new modes of action that have been released here recently, one for use in rice production and the other one a dual mode of action that should be used for broadleaf weed control in a range of broadacre crops, and that's just a recent one that's just coming online here. So some of our, I mean, during this kind of similar time period now in Canada, we've seen a pretty big change in some of our production systems, and one of those is the adoption of conservation tillage. Uh, Through one of our projects, we recently surveyed just over 1,000 farmers across Canada. You can see the number of farmers stratified by agricultural land base across the country. And this question was just asking when did those farmers adopt conservation tillage for those that did adopt it. And you can see the timeline here increasing largely throughout the 90s and plateauing here as of recent. During that same time frame, throughout the 90s and 2000s, 2010s, you can see that herbicide use patterns have changed as well. So this is from a report published by Alberta Environment and Parks back in 2020. And so they monitor the herbicide sales within Alberta. And so these are reported sales of different herbicide active ingredients. You can see the light blue line there is glyphosate. And you can see essentially tripling in glyphosate sales over the same time frame where we saw rapid adoption of conservation tillage systems. And so in this survey, it was a really large survey. I'm just pulling out a few interesting questions here. But this is one where no-till farmers, so there were 422 no-till farmers, and they were asked what factors are positively impacted by their tillage system. And you can see farmers report a whole range of soil related, economic related, as well as a few crop related factors that are positively impacted by their tillage system. You can see in dark green here is what they report is the greatest impact. Sort of the mid green is their second greatest impact, and then light green is their third greatest. And so what we observed here is that farmers report positive impacts of no-till on soil moisture, reduced erosion, as well as increased organic matter within their fields, and a whole range of other agronomic factors and soil-related factors that are positively impacted by no-till. As well, no-till farmers considered any pest-related factors as being impacted positively by no-till, which I thought was quite interesting. Now, if we flip that around and look at factors that farmers report being negatively impacted by the adoption of no-till, you can see at the top there, herbicide resistance is number one. The greatest negative impact of no-till systems on soil temperature and herbicide resistance, as well at number three there is weed management. So you can see that farmers are experiencing negative impacts of no-till on their weed control, and in particular the evolution of herbicide-resistant weeds. So this is creating a challenge, and it's one that we obviously need to address because we do need to maintain some of those positive benefits that our no-till systems have across the prairies. So our research program focuses on what's called biovigilance. And so this can be a bit of a complicated topic, but essentially what it is it's a research continuum looking at understanding, first of all, awareness of a particular issue, looking at detecting that issue, assessing it, so using some monitoring to assess what's the impact of it, trying to understand its biology and ecology, how do you mitigate it, and then were those mitigation practices appropriate or not? And this is essentially just, it's a research continuum that can be applied to any pest discipline. If we simplify that a little bit, essentially what we do is we identify threats to our production systems, understand their biology and ecology, use that to inform targeted management, and then reflect on whether those actions were appropriate or not, or potentially even causing unintended effects as well. So some of our research, one project or activity that I have the pleasure of leading under the Integrated Crop Agronomy Cluster is the establishment of the Prairie Weed Monitoring Network. So I'm one of the co-leads of this network, and some of our research activities here that we're doing between 2023 and 2028 are weed abundance surveys that are led by Julia Leeson out of Saskatoon, looking at weed density by species in over 4,000 fields across the prairie provinces. As well, herbicide resistance surveys conducted both pre-harvest and post-harvest in 800 fields across the prairie provinces over 4 years. Which is one activity that I'm leading under this project. And then also using some value-added modeling of the historical data sets on weed abundance and resistance to identify herbicide resistance risk for different unique herbicide-resistant weeds that may be at risk of evolving in the region, as well as how weed communities may change in response to management or climate factors. So a fairly large undertaking. And so this builds on some of the past research. So the herbicide-resistant weed surveys were initially led by Hugh Becky out of Agriculture and Agri-Food Canada in Saskatoon. And during the last round, they transferred over to my research program. but we're collaborating widely with weed scientists and specialists across the region. The most recent completed round of these surveys took place between 2019 and 2023 and covered 827 fields across the region, where we screened over 3,000 samples with multiple different herbicides to look at the impact of herbicide resistance in the region. And so what we do in these surveys is we visit, first of all, they're randomized surveys, so randomized selection of quarter sections, stratified by agricultural land base across the region. And we get permission to access all of these fields from the landowners. And so we visit the fields right before crop harvest. It has to be late enough that the weeds have mature seed, but early enough that the field isn't harvested. And we walk a W transect pattern within those fields and look for visible uncontrolled weed patches. And so we visit those weed patches, estimate their area, sample mature seed from those patches, and bring those seeds back to our center where we screen them for resistance using whole plant bioassays. And so aggregated information from these surveys, show the increasing percent of fields where at least one type of herbicide resistance occurs in each of the prairie provinces. Currently, we're sitting at about 70% of all fields under annual crop production have at least one type of herbicide-resistant weed present. Where resistance is found, it covers about half of the acres within that field. And when we aggregate this information together based on the estimated area of patches, we estimate that about 24.5 million acres across the region have uncontrolled herbicide-resistant weeds present at harvest time. So when this is combined with farmer-reported estimates of the cost of herbicide resistance in reduced crop yield and quality and increased weed management practices, we estimate that herbicide-resistant weeds cost prairie farmers about $658 million a year. And so there's quite a few different herbicide-resistant weeds that have been reported across the region. This is one of the fact sheets that has been developed by the Prairie Weed Monitoring Network, identifying various weed species and the herbicide groups to which they have been reported resistant in the prairie region. You can see that there's a large number of group 2 resistant weeds, but of course, there's resistance to a whole range of different herbicide modes of action. In these surveys, here we're looking at the percentage or number of fields from which each species was collected. So this isn't necessarily resistant weeds, but it's the greatest number of samples that are being collected. And so for broadleaf weeds, the most commonly collected species during the pre-harvest surveys are kochia, lamb's quarters, wild buckwheat, Canada thistle, and spiny sow thistle. For grass weeds, no surprise, we have wild oat as number 1, followed by green foxtail, barnyard grass, foxtail barley, and yellow foxtail, which is particularly problematic in southern Manitoba and just moving into southeastern Saskatchewan. So I mentioned that when those seed samples come back to our research center, we screen them using whole plant bioassays. And essentially what that means is we thresh out the seed. We plant the seed in a soilless medium in these trays in the greenhouse, and we grow them up and we treat them with herbicides using a moving nozzle cabinet sprayer. And you can see here just an example of a bioassay for glyphosate resistance in downy brome, where you have your susceptible control sample, you have the sample that you're screening, and then a resistant control sample. And essentially you just look at the phenotypic response of those plants to the herbicide. You can see at a broader scale here, we're looking at, this was a screen for glyphosate resistance in kochia, each tray representing a different sample that was collected. And this is just an image of the setup in the greenhouse for screening. So this, when it's done for the surveys, it's the same as when a farmer would submit a sample to a diagnostic lab. Essentially what they're determining is percentage resistance incidence, or basically plant survival in response to that herbicide. And depending on the lab, some labs give back a broad grouping of susceptible versus low, moderate, or high resistance. Others give you back the exact percentage. Um, I find that there's too much focus on the actual percentage. These are very broad estimates, and I would caution anyone away from focusing too much on the percentage. If it's greater than 20%, it's quite likely going to manifest as lack of control in a field. But changing, say, 60 to 70% is something that you probably shouldn't be considering in your decisions. It's a pretty broad estimate. So this is just showing the various herbicide-resistant weeds that were reported for the pre-harvest surveys, which predominantly focus on Group 1 and Group 2 herbicide resistance. And so you can see here, this is the percentage of tested fields where resistance was reported. And what I mean by that is tested fields are the number of fields from which the species was collected and tested. So overall, we surveyed 827 fields. We collected and tested wild oat from 441 of those fields. And out of that, it was about 80 to 85% of them had group 1 resistance present. So that's how you would read this for the whole range of biotypes. As well, I mean, green foxtail, we're sitting at about 50% of the tested fields across the region having group 1 resistance, and yellow foxtail, pretty close to that, but just under. For group 2 resistance, essentially all kochia populations tested in recent decades have had group 2 resistance, and there's a whole range of other weed species that vary in their resistance to group 2 herbicides. For group 1 and 2 multiple resistance in wild oat, we're sitting at about 45-ish% of tested fields having both group 1 and group 2 resistance across the region. This is perhaps a better way to look at it, or the way that I like to look at it. This is the percentage of all fields across the regions that were surveyed that have these types of resistance present. So regardless of whether the species was collected or not. So for group 1 resistance in wild oat, 46% of fields under annual crop production in the region have group 1 resistance. Green foxtail was at 11% for group 1 resistance, and yellow foxtail at 3%. Group 1 and 2 resistance in wild oat in about a quarter of the fields across the region. For kochia, was the most commonly identified group 2 resistant weed at 30% of fields across the region, followed closely by wild oat at 27%, spiny sow thistle at 10%, and then a whole range of other grass and broadleaf weeds that are at either 5% of fields or less. Now for kochia and Russian thistle are sampled separately in a post-harvest survey, just simply due to the timing of seed maturation and viability within these species. When we sample them from lateral branches beneath the combine height, post-harvest, we tend to get a better seed sample for resistance testing. So I mentioned group 2 resistance in kochia was tested in the pre-harvest survey, and then we test for group 4, group 9, and now group 14, as well as some additional targeted testing for group 5 as well in survey samples that are collected post-harvest. So the post-harvest surveys, or at least the most recent completed round across the prairies, took place between 2018 and 2021. And essentially what we found was that about 3/4 of the samples that were collected and tested had glyphosate resistance present, and just under 1/4 had dicamba resistance or group 4 resistance present. Fluroxypyr resistance is another group 4 or auxin mimic herbicide, and it was tested only in Alberta because it was, it was for research interest but unfunded, and what we found in Alberta was that 44% of those samples had fluroxypyr resistance. So we're seeing greater resistance to fluroxypyr than we are dicamba across the region. In addition, now we also have group 14 resistance across the region, which I will talk about in a bit here. So in addition to the surveys, my research program also offers free testing for grower-submitted samples, but only for those samples that suspect novel resistance. So this means the combination of a herbicide mode of action and weed species for which resistance has not been documented across the region. And so each year we end up getting samples of suspected novel resistance, and we do some of the initial research to characterize new types of resistance in the region. And so this has resulted in the first confirmation of 5 new herbicide-resistant weeds in the last 5 years. Glyphosate resistance, or group 9 resistance in downy brome in 2021, group 14 resistance in kochia in 2021, group 1 resistance in foxtail barley in Alberta in 2022, group 2 resistance in Japanese brome in Saskatchewan in 2022, and group 2 resistance in corn spurry in Alberta in 2025. So I'll touch on each of these here quite briefly. So glyphosate-resistant downy brome came in from a Roundup-ready canola field in the MD of Taber here in southern Alberta back in 2021, where the farmers reported lack of control with glyphosate applied both multiple times pre-seed and then also 2 in-crop applications as well. And they still didn't have control of the downy brome within that field. So they submitted the sample to our lab, and we conducted a dose response experiment here. Essentially what this is it's exposing the putative resistant population shown in the back here, compared with 2 susceptible populations in the middle and the front, exposing that to a range of rates of glyphosate to see how it responds. And a rough field rate highlighted there, what we found was that indeed the downy brome had evolved glyphosate resistance, and we have gone on to report the mechanism, which is similar to the mechanism of glyphosate resistance in kochia. So far, glyphosate resistance in downy brome has not been reported beyond this initial municipality where it was first documented. Group 1 resistance in foxtail barley came in from creeping red fescue fields in the Peace River region of Alberta back in 2022, where they suspected Quizalofop resistance. You can see similarly the dose response showing that the resistant population surviving field rates of Quizalofop. And we have gone on to also report the mechanism conferring resistance in this species as well, and used a genetic test to identify it in further fields in the Peace region, but not beyond that region to date. Group 2 resistance in Japanese brome. So this is samples that came in 2022 from 2 Durham fields in southern Saskatchewan. And you can see a photo of the dose response experiment there, where we're seeing very, very high resistance up to and greater than 100 times the field rate of the herbicide. And so this is also one that we have not found beyond these initial cases, at least to date. Group 2 resistant corn spurry is the most recent one that was submitted from a canola field in 2025. And this is also the first known case of this, and it was from the Peace River region here in Alberta. And we've gone on to report resistance to a range of group 2 active ingredients within this species, and just recently identified the mechanism that's conferring this as well. So group 14 resistance in kochia, I have stay tuned here because this is an example that I'm hoping to use later on here in the presentation. So some of our other work, I mentioned reporting mechanisms of resistance. And so some of our work aims to document what are the actual genetic mechanisms that cause herbicide resistance in these various weed species. And many people are surprised to learn that there's multiple different mechanisms in plants that can confer herbicide resistance, broadly categorized into target site-based resistance and non-target site-based resistance. And both of these do occur in weed species on the prairies. And so I'm not going to go into detail on all of these mechanisms per se, but I will focus on target site resistance. And one thing I did want to point out is that this research to identify mechanisms is leading to the development of genetic tests to rapidly characterize and identify resistance. And I'll touch on that here in a bit. But they target mainly these target site resistance based mechanisms, while whole plant bioassays are nice because they work regardless of the mechanism. So I'll talk about target site mutations because these by far are the most common type of mechanism that confer herbicide resistance among weed species. And so essentially the way this works is you have your herbicide active ingredient and you have an enzyme binding site. So that the herbicide active ingredient binds to the target protein, and successful binding means that it is impacting part of that pathway that is necessary for successful growth and development. And so in reality, it's not as simple as the simple diagrams that I can make in PowerPoint here, but it's quite a bit more complicated than this. But this is just an image of a model that's showing clethodim binding to the ACCase binding site here, just to show you that it is more complicated than I'm describing here. But in the case of a target site mutation now, you have your active ingredient and there's a genetic mutation that's resulting in a structural alteration of that enzyme to which the active ingredient binds. And when the active ingredient goes to bind, it cannot bind as successfully, and it has what's called reduced binding affinity, meaning that it's not having the same activity on that critical pathway. And so we're characterizing the mutations that are conferring target site resistance in various weed species, and also developing genetic tests that we can use to identify this rapidly during the growing season. So under one of our recent projects, we developed this genetic testing kit where essentially you can collect a small amount, about a 3 to 4 inch size of leaf or of seedling tissue, and place it in the Ziploc bag that's filled with silica gel beads, and those beads just desiccate the tissue while the sample's being sent back to the lab. And when it comes to the lab, they extract DNA from the plant tissue, they characterize it, and we, in our research behind this, we sequence the target site gene. So this is a partial sequence of ALS in cornsbury, documenting one of the mechanisms that we reported in cornsbury that is resulting in an amino acid substitution that causes a structural change in that target site enzyme. And so this is all used to develop a genetic test that we can use to separate resistant from susceptible plants. So we've developed this pathway to characterize resistance by identifying new types of resistance and extracting their DNA, sequencing the target site gene, and designing a genotyping assay. And so once we get through the research and characterizing the mechanism of resistance, we can jump straight to this third step where basically you collect the sample, you extract the DNA, run a PCR, and you have a result. And so what this means is that you can get a result more rapidly than planting the seed and going through spraying it and looking at the phenotypic response. So over the last about 5 years, we've characterized mechanisms conferring resistance in 17 different unique herbicide-resistant weeds across the region. using this to develop a portfolio of genetic tests that can be employed. And this is all developed based on weed issues in Western Canada. So if we look at the various herbicide-resistant weeds that we have here, so far we've developed genetic tests for the ones highlighted in red there, and we're continuing this work on to further characterize mechanisms of resistance in other weed species. So this is the value proposition for genetic testing for herbicide resistance. So we have basically 2 calendar years here in a timeline. So traditional whole plant bioassays, if you in year 1, so you're applying your herbicide, say in June, for example, scouting the field a few weeks later and you notice lack of control. Well, unfortunately, you can't do anything about it if you're doing traditional whole plant bioassays. You don't have confirmed resistance until you let those weeds go to seed. You collect the mature seed and you submit it to a lab, and that lab plants it in the fall or in the winter and screens for resistance using a bioassay over the winter, and it can take a few months to get a result back. The genetic testing, though, is more rapid because as soon as you note lack of control in the field, you can sample leaf tissue, submit it to the lab, and they can run these tests and have a result back to you within about 1 to 2 weeks. So what that means is that it can inform alternative management practices within the same growing season rather than the subsequent growing season. So here we're just comparing and contrasting the 2 methods. So genetic testing requires leaf tissue, whereas bioassays require mature seed from the weeds. We must know the mechanism for genetic testing, and whole plant bioassays work regardless of the mechanism conferring resistance. So genetic testing is based on target site resistance mechanisms, and it's much more difficult to create these for non-target site based mechanisms, whereas bioassays work regardless of the mechanism. I mean, the lab space that you require for each is different as well. Genetic testing is plant level diagnostics, whereas bioassays are more population, so you get that percentage value. But the biggest difference here is the timeline. So 1 to 2 weeks for genetic testing, whereas whole plant bioassays can be a few months before you get a result back. So let's talk about a case study here now. So this is group 14 resistance in kochia, a new issue that's showing up in the region and unfortunately seems to be spreading quite rapidly. So here's the timeline of herbicide resistance in kochia. So many of you are probably familiar that kochia can exhibit resistance to multiple different modes of action. Up to 5 different herbicide modes of action have been reported globally, 4 of which are known to occur in Canada. You can see group 2 resistance in Canada showing up in the late 80s, followed by glyphosate resistance here in 2011. Then auxin mimic resistance, so this would be dicamba and fluroxypyr resistance showing up in 2015. And then more recently, PPO inhibitor group 14 resistance showing up in 2021 in Canada. So we recently published a paper that highlights the first report of group 14 resistance in kochia globally. And we worked with our colleagues out of North Dakota State, where we had identified, an agronomist in Saskatchewan identified that a sample that may be group 14 resistance and sent it to our lab back in 2021. And then our colleagues in North Dakota observed something similar there in 2022 as well. So we were kind of working on this together. And here just showing a dose response to 2 group 14 herbicides. This would be saflufenicil and carfentrazone. And some of the results from that dose response experiment here, you can see the differential response over a log scale for these 2 group 14 herbicides, showing that our resistant population exhibited between 50 and 100-fold resistance to these various group 14 herbicides. So here we're highlighting the various active ingredients within group 14, and not all of these are registered for use in Canada. But we have gone through similar experiments looking at resistance to various commonly used group 14 herbicides in the region. And we've unfortunately found resistance, both soil applied and foliar applied resistance to a range of group 14 active ingredients from different families within group 14 as well. Interestingly, though, it appears that these diphenyl ethers, so the family called the diphenyl ethers, continue to work on group 14 resistant kochia, which is quite interesting. Unfortunately, the use cases for most of these herbicides, the ones that are used in Western Canada, are primarily based in Manitoba in soybean production, as well as some dry bean production in the Red River Valley as well. And there's a few horticultural uses too. So here, there's a lot going on this slide, but this is assuming that you have group 2, 4, 9, and 14 resistance in your kochia population. What herbicide options are left in the various crops commonly grown across the prairies? And so the green with a C would mean that these herbicides are labeled for control, whereas the S would be suppression. But you can see that we're starting to see weak links for when it comes to the stacking of resistance traits in kochia, especially when we're talking about post-emergence. I mean, this isn't a new issue, but this is primarily because of group 2 resistance. So post-emergence, there's very few, if any, options in our pulse crops. But also, we're starting to see a weak window here also looking at pre-emergence in some of our cereal crops as well. So you'll see there is some labeled for suppression, but if you look at the burn down scenario, we're looking at relying, well, burn down and post-emergence, we're relying on bromoxynil either alone or applied in combination with a group 27 in essentially all of both the burn down and the post-emergence windows. So a lot of reliance on group 6 and 27 there. As well in canola now, there'll be quite a bit of reliance on group 10 or glufosinate post-emergence, which is something that hasn't changed but is top of mind, I think, for resistance selection. So for group 14 resistance in kochia, we had conducted a baseline survey from those samples that were collected back in 2018 to 2021. And across 882 samples during that time frame, we did not find group 14 resistance in any of them. But during a similar time frame, 2021 to 2023, we did confirm this in 2 grower-submitted samples. The first one is that initial case from West Central Saskatchewan that I spoke about previously. And then we also identified a second sample in Southern Alberta in 2023. Moving forward, we've recently submitted a paper here documenting the novel mechanism of group 14 resistance in kochia alongside colleagues from BASF and North Dakota State as well. So we do know the mechanism at the genetics level that's causing group 14 resistance in this species. And based on that, we designed genetic tests to rapidly confirm group 14 resistance. And so these slides will just show you what's possible with the genetic tests and how quickly we can get an idea of how widespread group 14 resistance is. So we used survey samples that were collected from Saskatchewan in 2024 and screened them all using bioassays in the greenhouse. And then any surviving plants we tested using our genetic testing protocol. And so you can see here that we confirmed group 14 resistant kochia now across both the mixed grassland and moist mixed grassland ecoregions within the province. So overall, across the 205 fields that we surveyed here, 28% of them had the mechanism present that confers group 14 resistance in kochia. And if we look here, separated in based on plant survival, you can see if you look at the fields tested here, you can see that 6% of fields had greater than 20% survival, meaning that this would be visually obvious as lack of control in the field, while 22% of those were developing resistance, meaning that the mechanism was there, but it was still spreading within the population to a level that would probably be visual as lack of control. So it's an issue that is developing quite quickly among these kochia populations in Saskatchewan. Moving forward here, we also deployed this genetic testing kit across the region this year. And simply, I just made a call over X saying that we have this genetic testing kit, and we can test samples for free just to validate our genetic tests that we developed last spring. And so overall, we ended up getting 891 plants submitted by farmers and agronomists across the region, representing 91 different fields across the region. And the mutations conferring group 14 resistance in kochia were documented in 26% of those fields. This is just showing the overlap of different target site mutations that confer group 14 resistance in kochia. But what we did was we also documented it in southern Alberta and also southern Manitoba as well. So now combining these two surveillance efforts together, we come up with the current map of group 14 resistance in kochia across the region, shown here. And to put this into perspective now, we had 2021 to 2023 samples shown in blue, and then the genetic testing that we deployed just during 2025, as well as those 2024 survey samples. And you can see how quickly we're getting an idea of where group 14 resistance is occurring in kochia populations across the region. So we also do a whole range of small plot research looking at integrated weed management strategies targeting these various different weeds. Unfortunately, I don't have time to get into a lot of this today, but this is something that both my colleagues will be also talking about here in the subsequent presentations. In general, the idea is to push away from our contemporary low diversity weed management programs and integrate both basically physical, cultural, and biological weed management practices to increase the diversity of our weed management portfolio and help to mitigate further selection and spread of herbicide resistance. So in conclusion, I mean, it's pretty clear actually that contemporary weed control is failing due to lack of diversity. And so what I mean by this is an over-reliance on herbicides, and there's a dire need to increase the diversity of our weed management programs to help avoid further selection of resistance. About 70% of fields across the prairies have herbicide-resistant weeds uncontrolled at harvest time. We estimate this costs farmers in the region $658 million a year. There's 5 new herbicide-resistant weeds in the last 5 years in the region, so just new weeds to watch out for. And what I talked about today as well is we're working on characterizing mechanisms of resistance and developing genetic testing that is a tool that may be available at diagnostic labs as early as 2026 or 2027. So with that said, I'd like to thank everyone for listening. Our research on herbicide resistance is supported by a wide range of funders across the region. And of course, it's not possible without all of our collaborators and the dedicated staff and students that help make it possible. So thanks everyone for listening. Happy to answer questions.
Thanks so much, Charles. It was a great presentation. And I think those maps really, give us a visual of how far across province those problems actually are. Our first question here is, will there be free resistance testing for 2026, and how can a producer or an agronomist submit samples?
So yeah, excellent question. So the resistance testing, it changes from year to year. So we'll still be offering free testing for grower submitted samples, especially these will be the seed samples that with suspected novel herbicide resistance. So if you go on the Prairie Weeds website, so prairieweeds.com, there's a fact sheet there that identifies the various herbicide resistant weeds that occur in the region. So anything that's not on that fact sheet is something that we will test for free using mature seed samples and bioassays to start off with anyway. The genetic testing, there will probably be some more targeted sampling with the kits, but likely not group 14 resistance because we've confirmed that those tests work very well. And it will depend on whether there are commercial labs that are able to offer these tests or not. So we're working on licensing agreements with different labs that will hopefully offer these tests as a service to farmers. So if they're offering it, then we won't be because we work on the development side of things.
Great, makes sense. Next question. If there is fluroxypyr resistance, showcasing the small research trials, would it be better to use this group 4 along with another mode of action instead of adding more of the active ingredient?
I mean, the answer is yes. So essentially, yeah, I mean, with fluroxypyr resistance, we have an idea of what the mechanism might be, and ideally, rather than adding more of the same herbicide, what you should be doing is using an alternative mode of action along with that. I mean, of course, integrated weed management principles, right? It's not just herbicides, but I mean, using other effective modes of action as well. Some of our other research has shown that there is synergy among oxinic active ingredients as well, and so it's if you have, say, fluroxypyr resistance, it doesn't necessarily mean that you have resistance to those other auxins as well. So I'm saying other modes of action, but other auxins mixed in there may also be a short-term management practice as well.
Perfect. The next one is, did you notice any phenotypic differences in leaf shapes, size, and appearance on new types of resistance versus susceptible leaves of kochia?
We've noted it a little bit. We have a data set that will be actually have to analyze it for a report here coming up shortly. But we've been looking at the fitness cost of different herbicide resistance traits. And so in some cases, when you have, say, a mutation or a mechanism that causes resistance, that comes along with other changes as well. These are called pleiotropic effects of herbicide resistance traits. But essentially what that means is that there can be other alterations within the plant that impact the plant's ability to survive and reproduce and contribute to subsequent generations. Some of that can be a change in leaf shape, leaf angle. I mean, there's a whole range of things. I mean, seed production is a big one that we look at. And so it really depends on the weed species and the mechanism that's causing resistance. So it's hard to say at a broad scale, but we've noted some of that in kochia, yeah.
Fair. Do you think the level of resistance development could depend on abiotic weather parameters in relation to seed production versus susceptible population?
You might have to read that one again, sorry.
Not a problem. Do you think the level of resistance development could depend on abiotic weather parameters in relation to seed production versus susceptible population?
Yeah, I think I know what that question is getting at, but there's definitely an interaction between abiotic factors, right? So, um, I mean, at a broad scale, resistance is related to stress, right? And so there, so abiotic stress that as a result of weather could potentially result in faster or slower development of resistance, depending on the mechanism, though. So it's really hard to say yes or no clearly because it really depends for that situation.
Okay, next one is there a genetic test for wild oats?
There is a genetic test for wild oats, and so the there's a bit of a so the test is available, but it is also only telling a small part of the picture. And so what I mean by that is that we know that herbicide resistance, especially group 1 and group 2 resistance in wild oat, can be conferred by multiple different target site mutations, which these tests can pick up, but also metabolism-based resistance in wild oat, which the genetic tests currently do not cover. So you're missing the impact of enhanced metabolism on resistance there. In addition, the biology of wild oat is highly complicated, especially because it's a hexaploid species. And you can have target site mutations on essentially some of so there's the different ancestral genomes in a hexaploid species, and target site mutations can affect, say, one of those or multiple. And you have this dilution effect where you still have you have resistant alleles and susceptible alleles as well. And so the resistance that you see in response to a target site mutation can still be anything from complete control to complete lack of control and everything in between. So while there are genetic tests that can pick up a target site mutation, you're missing a lot of the practical application behind it. It'll say, yes, there is a mutation, but then what does that mean at the phenotypic level? What does that mean for control in the field? That link is very, very muddy. So I would suggest if you're looking at resistance in wild oat, at least currently, until we can further research in that area, I would suggest sticking with a whole plant bioassay.
Perfect, makes sense. And I think that's going to wrap up your question period so we can move on to our other two speakers. So thank you so much again for taking time out of your day to come here and present. We really appreciate it.
My pleasure. Thanks for your interest.
All right,
Take care.
thanks. Have a good day. All right, next up, we have Dr. Breanne Tidemann. Breanne is a research scientist with Agriculture and Agri-Food Canada in Lacombe, Alberta. She grew up on a grain farm in West Central Saskatchewan, but completed all of her degrees at the University of Alberta. She holds a bachelor's in biological sciences and a master's and PhD in plant science with projects focused in weed science. Breanne started with Agriculture and Agri-Food Canada in 2016 as a weed scientist. Her research program in Lacombe focuses on integrated weed management strategies and novel weed management practices, as well as weed biology and ecology. Okay, your presentation looks like it's ready to go. Go ahead, Breanne.
Perfect. Thank you so much for the introduction. So Charles has set us up really well in terms of sort of what is happening in terms of herbicide resistance. I'm going to go a little bit more into some novel management strategies that we're trying to work on, and then kind of circle back at the end to a little bit more herbicide resistance work that we're doing. Before I jump into that, I just want to take a moment to thank the staff and the students that I work with. They're the ones that are out trying to count how many wild oats are showing up in wheat or barley and probably cursing me as they do so, yet they're still here and still helping. And so I very much appreciate their time and efforts, as well as a very big thank you to all of the funders of my research program. I'll reference a few of them throughout the presentation with specific projects, but all of their support over the years has been incredibly appreciated and gives me the ability to do some of the work that we're going to talk about. So again, as I mentioned, Charles has set us up really well with the current situation in terms of we are really seriously dealing with herbicide resistance, and it brings us to a place where we can't rely just on new herbicides to solve the problem for us. And so the question that we get is, what else should a producer be doing? So we're playing in a bunch of different areas trying to figure that out, and we're going to go through a couple right now. So I want to start by talking about harvest weed seed control. What is harvest weed seed control? It's a paradigm of weed control that was developed and refined in Australia. And the goal of harvest weed seed control is to manage weed seeds that are still in the field at harvest and prevent them from being dispersed back across the field and getting into your seed bank. So we're trying to stop inputs into the seed bank. Our combines are really great broadcast seeders in that a lot of our weed seeds that are still in that field will go into the combine, be separated into the chaff, and then our spreaders do a great job of spreading that residue, right? So we're just broadcast seeding them back across the field. Really, we want to quit enriching that seed bank because that seed bank is an issue for us for multiple years. Now, I do get a lot of questions or a lot of comments that, doing this is not going to save our yield because we've already had those yield losses from the weeds, and that's correct. What we're trying to save is now next year's yield or a few years down the road if you're dealing with a longer term seed bank like wild oat. So we're trying to protect future yields. We're not really worried about what's already happened that season. In order for harvest weed seed control to be effective, those weed seeds still need to be on the plant when we're harvesting the crop. So if we've already dropped those weed seeds onto the ground, they're already gone. We're not going to get them back. We're not going to be able to get them into the combine. So that's kind of, criteria number one. Number two is that they have to be produced at a height where they will be collected by the combine. So we need them up a little bit in the canopy, or you need to lower that cutting height lower to the ground. The third thing is that you need to be able to collect the weed with the combine, which I've pretty much just told you that you can do that. However, there are some weeds, and these are two that I saw in Australia. They're very round, they're very bushy. And so when we ran a combine through, rather than feeding into the combine header, they rolled up onto the header and then off the sides. And so while they had really great seed retention and the seeds were more than produced high enough in the canopy, we couldn't get that weed into the combine. So I had to add a third criteria here on my list. Now, the method that I'm going to focus on today of harvest weed seed control is impact mills. This is a type of mill that's attached to the combine and it processes the chaff. So in this top right-hand picture, you can see rocks or coal going in here. This is what these types of mills were traditionally used for. And you see big pieces going in and small pieces coming out. And that's the entire idea, is the chaff would go in, it would be milled and broken down with much smaller pieces coming out. And then that residue goes back onto the field. So if you look at this image of one of the early prototypes of these impact mills, the straw fraction that comes through your harvester is not touched. We're not touching the straw. We're only focusing on the chaff, and that chaff gets fed into a mill, and then those residues, both the straw and the chaff, do go back onto the field. So we're not messing so much with that nutrient cycling, but we are trying to target the viability of those weed seeds. Now, on the left-hand side is actually the impact mill version that I've got to work with. This is the original tow-behind Harrington seed destructor. As you can see, it's a rather large, bulky, not super convenient type of unit to have behind your combine. However, we have seen technological improvements, and now we're looking at impact mills like what's on the right-hand side. So these are built into the backs of the combines. There's no towing anymore. It's right component of the machine as it's going through the field anyways. So there are 4 brands of impact mills currently on the market. This bottom right picture is the IHSD, or the Integrated Harrington Seed Destructor. So it is the direct evolution of the one that I got to work with that was towed behind the combine. On the bottom left is the Seed Terminator. On the top left is the Redicop Seed Control Unit. Now, Redicop is based in Saskatchewan. There are only Canadian brand of impact mills to date. And Tech Farm Weed Hog, which is just sort of coming onto the market, is another version that's coming out with a slightly different mill system, but the same concept. So the Weed Hog, the IHSD, and the Seed Terminator are all built in Australia. Again, the Redicop is the only actually non-Australian version available on the market. Now, we've focused in on impact mills in Canada. There are other methods of harvest weed seed control. However, we believe impact mills to probably be the best fit for most farmers in Western Canada and really to the east as well, although individual farms will of course vary. Why do we like them? They can be integrated into the combine. Unlike some other harvest weed seed control methods, it doesn't require fire or lighting things on fire, which is always a good thing. And it does leave that residue on the field while it's actually doing something to the weed seeds. So those are sort of the reasons that we've focused in on this particular strategy out of the harvest weed seed control options. A little bit of data in some of the work that we've done, starting with sort of those 3 criteria. So seed retention, weed seed retention is species dependent. So on the left-hand side is a graph of 3 different weeds. We've got wild oats in green, we've got cleavers in red, and we've got volunteer canola in blue. And you can see a timeline sort of through August and September in this case. And you can see wild oat is starting to drop its seeds very early. Cleavers holds on a little bit longer before it starts to shed those seeds, and then volunteer canola held its seeds the longest out of those 3 species. And so what we see is that what that retention looks like depends on what species you're talking about, and that retention does differ by environment or year. So if we have a really dry year, if we have an early snowfall, if we have a lot of wind, we'll see differences in that seed retention. The other component is that it is cropping system dependent, and I'm going to show you that a little bit as well. But basically, if you have a later maturing crop, you're probably going to see more or less seed retention by the time you harvest, more seed loss, because those plants are just out there growing for longer, right? So the longer and the later you're going to harvest, the more seeds you tend to be dropping. So if you're an early maturing crop, say lentils in Saskatchewan, you're probably going to see more seeds still left on those weed plants. If you're a later maturing crop, maybe faba bean, something like that, you're probably going to have less seeds on that crop. So cropping system and where that harvest timing falls also comes into play here. Now, we also did a study looking at cropping systems, so a 3-year rotation where we had early, normal, or late crops. So a normal was a canola wheat rotation, and early was a pea and winter wheat, and our late was faba bean and flax, if I remember correctly. And so over 3 years, we sort of had these differences in cropping maturity. And then we also layered in looking at swathing and straight cutting. And what I just want to emphasize here, this is looking at wild oats in the 3rd year, which was all in barley. So the impacts of the previous 2 years are really showing in our wild oat densities by the final year. And we have a much lower wild oat density in our early maturing crops because we were getting to those wild oat seeds a lot earlier. Plus we had a really great winter wheat stand, and we know winter wheat is competitive against wild oat. So there was sort of a double stacked impact there of the early maturity and the improved competition on the wild oat. So that timing matters, those crop choices matter for helping target weeds with this particular strategy. What does it look like after something goes through an impact mill? This is 3 different kinds of chaff. We've got barley chaff on the top left, pea chaff on the top right, and canola chaff on the bottom. In all of these images, the left-hand side is what our chaff looked like when we put it through. You can see a couple pieces of straw and things like that where it wasn't pure, but it gives you a very good idea of what might be going through on that chaff stream. And on the right-hand side is what was left after it came out of the impact mill. As you can see, it's very fine, it's very powdery, it's almost flour-like in texture. It gets under all of your masks and any hearing protection you're wearing and along the cuffs of your shirts and gets very, very itchy. And yes, that's from experience. But you can see it's really reduced the size of that material. And so you can imagine that any wheat seeds in there have also been broken down into smaller pieces. And when you break them down into small pieces, they're not able to germinate and then be a problem in your field. I'm not going to go through all the different research that we've done on this. I'm just kind of going to give you the takeaways from it. If you can get it into the impact mills, you're typically looking more than 95% kill of those weed seeds. And in many cases, it's greater than 99%. That 95% is averaged across not only the Canadian work that we've done, but sort of across all of the work that's been done in the United States and Australia as well. There's very few weeds where we're seeing under 97, 98% control once we get them into the impact mills. Now, we do see things in terms of looking at weed seed size or looking at weed seed number, looking at chaff volume or chaff type, where some of these things will impact control. However, it's such a small fraction of a percent in a lot of cases that, yes, statistically we have significance, but biologically it doesn't really matter. If you can get it into those mills are doing a very good job. The bigger trick will be getting it into the combine and dealing with those seed retention challenges. Okay, so if you can get it into the mill, they do a great job. Can you get it into the combine? To get it into the mill is the bigger question. Now, these are images of very not happy mills. This is the #1 challenge that's been reported by our early adopting farmers in both Canada and the US. And what's happened here is there's been too much tough or green material that has gone through the mills, resulting in plugging. Okay, so a really big patch of green kochia, soybean straw that still has some green left in the stem. These are the types of situations where we're seeing this happen. The mills don't like tough material. Okay, they're trying to make flour, and when it's got a lot of moisture in it, then things start to stick and then they start to plug. That's essentially what's happening. So if you have a very, very, very large patch of green kochia and you're trying to harvest a wheat crop in it, the recommendation is you still want at least 50% of the material, if not more, going through be dry material to help push it through. If you can't get that proportion, then you're probably going to want to go around that patch. You're going to want to either desiccate it, you're going to want to swath it to dry it down, or you're going to want to leave it till it matures on its own and come back and clean it up like a slough. Chances are, if your kochia is that dense and the patch is that large, you probably don't have a great cereal yield in there anyways, and the time you're going to spend unplugging your mill is probably not worth what you're going to have collected in your actual grain hopper. So just to be aware of, this is a challenge that some of those producers are dealing with. There are ways to manage it. They are managing it. These pictures are from when they took a chance and lessons were learned, I guess you could say. A couple other things to know about impact mills. They do cost right around 100 grand. Okay, that's give or take where we're sitting. I do get pushback on that is a very large expense, which I do understand. I guess my challenge is how much would you push back on a precision sprayer or a targeted sprayer, which is still relying on herbicides for management versus a completely novel management strategy in your cropping system, right? So trying to take a little bit of a different perspective there. We are beginning to see adoption. There's an estimate of about 40 to 45 mills running on the Canadian prairies, anywhere from sort of 2,000 acre farms up to more than 10,000 acre farms and everything in between. There are challenges with them. I just showed you the plugging, and there are some strategies needed to deal with them, but they are developing and being looked at. And we do have an interest in the spatial benefit. So a lot of times we focus on how much can it drive down those weed numbers, right? But there's a spatial component to this too. And I just want you to use your imaginations with me for a moment. If this combine is going through a wild oat patch and it doesn't have a mill on it, as it proceeds through the patch, you're going to see wild oat seeds spread down the field. Even if there's not very many of them left on the plant because it's got poor seed retention, there will be some spread down the field, and this resistant patch that was maybe only the size of the combine has now grown in size on this field, right? If we use an impact mill and we destroy any of those wild oat seeds that are coming through the combine, can we keep this patch limited to right around the combine? We'll still probably see a little bit of spread because of running the seeder through there or just where those seeds have fallen beside those plants already. But maybe we can keep that smaller rather than spreading it down the field. Maybe that allows us to do some targeted patch management strategies on the field rather than having to really get creative with our management across the entire field. So that's sort of the concept. We've been working on this in a producer's field in Saskatchewan, and Shaun, who will be talking next, has been working with me on this. It's funded by the Sask Ministry of Ag and Sask Wheat. And we're actually seeing this occurring. So we're monitoring wild oat patches, and we're monitoring when the producer has not run his mill his impact mill in those patches versus the size of the patch if he is running the mill. Now you'll see both groups have seen an increase in wild oat patch size. Again, there's other ways those seeds are moving, whether it's with the seeder, whether there's some tillage happening, or just plants dropping seeds outside of their radius each year. But there's a pretty big difference between the growth where there's no mill versus where there is a mill. So again, it's helping contain those patches. Now, this is preliminary data. This is still an ongoing study, but just to give you a little bit of a visual that there is an impact of how fast those resistance patches are spreading as well. I want to shift a little bit and talk about a new project that I'm working on with something called directed energy. So this still falls under the harvest weed seed control idea. So we're still trying to manage weed seeds at harvest, but it's not using a mill that is actually breaking those seeds into pieces. So directed energy is patented technology by Global Neighbor Inc., who's an American company. They do have a commercialization partnership with Redicop. So the same group out of Saskatchewan that's developing the seed control units also is working with Global Neighbor on this directed energy idea. Now, the directed energy uses a combination of blue light and infrared wavelength light to eliminate weed seed viability. So this picture on the left is our benchtop version of what they call their weed seed destroyer. This is not what it will look like in a combine. This is just our benchtop testing version. So you can see there's a hopper here, and then down this tube, there's an auger that runs, and it exposes the weed seeds in the chaff to that blue light and that infrared wavelength light. Okay, we do have testing ongoing with a number of Canadian weeds. My students are actually running a trial as we speak. And then we're testing viability of all of these weed seeds after we have tested them through this particular unit. And we're looking at different levels of that infrared wavelength light to see where we're getting efficacy as well. Some very preliminary data. We are seeing reduced germination at higher temperatures, which would be that different level of the infrared wavelength light. That is coming through as heat, essentially. And it's really interesting to look at them because the seeds look structurally sound, but the internals of that seed are altered. So when we're testing seed viability, we do a germination test and see how many grow. Those that don't grow, we do what we call a press test or a squash test, sometimes you'll hear it called, and that's what's happening over on the right-hand side here. So the first seed and the third seed have also been squashed, but there was just nothing left in them. Those are not viable seeds. The second seed and the fourth seed are considered still viable seeds. So you can see that inside it's a fairly bright white color. It's still got a fairly solid or paste-like consistency. Those are viable seeds. What we're seeing in this study is when we're going to do our press test, those insides have essentially liquefied. Like they're gross. They are a little gross when we squish them, which is leading us to think that there may be something there that is actually happening with this treatment that is reducing our weed seed viability quite a bit. So not only are we seeing reduced germination, but we're changing something about the internal integrity of that seed that is also reducing the viability in addition to germination. So we're not just putting those seeds into dormancy or something like that. We are actually eliminating their viability. Now, again, this is super preliminary. My students have just finished sort of doing the first round of these tests and starting to squash the seeds. And I'll be looking at the data in the next couple weeks. But again, being over there with them while we were doing press tests, it was like, oh, this is a very different inside of a wild oat seed than I've ever seen before. So that was really interesting to see. The Directed Energy Project is funded by Saskatchewan Pulse Growers, as a side note, and it's been a really interesting experiment to get started on and kind of get a first look at some of this technology. So that's been a lot of fun. Now, the assumed benefits of directed energy, why this is of interest, why not just go with an impact mill? The idea here is that it will take less horsepower than the mills because you're not actually physically milling all of that chaff. The hope is that it will be a lower cost, although it isn't commercialized yet, so we don't know what that price tag is going to be. But the goal is that this would be more appropriate for older or smaller harvesters. So the mills themselves, typically you want to be running about a Class 8 combine, and the horsepower draw, essentially you're operating it as a Class 7. Less than a Class 8 combine, you start being pretty limited in terms of horsepower availability. So the goal here is to have a form of harvest weed seed control that would work on smaller combines, potentially older combines, which would be more suitable for some of our smaller acreage farms that maybe aren't buying the X9s right off the line and that type of thing, okay? So we're looking at these as sort of a complementary strategy for a different sector of our farmers. But again, it would have therefore an increased applicability of harvest weed seed control where more people could start using this strategy and start potentially incorporating it on their farm. So really interesting work, really new work, and still really interested to see where this goes and how this comes out to the market. The next thing I want to talk about is competitive varieties. This is a project that I'm just finishing up that's funded by Sask Wheat and Manitoba Crop Alliance. So a lot of times when we talk about integrated weed management, we talk about crop type and crop variety. And when we talk about crop type, there is quite a bit of information out there that's available on relative competitiveness of crop types. So we know barley and canola are often pretty close and can be a little weather dependent, but they're typically top of the line in terms of competitiveness, followed by wheat, flax, soybean, and then other pulse crops. With our pulse crops, we know they are kind of notorious for not being super competitive. But when we give these talks, when you look at the literature, when you look at discussions about IWM, a lot of times competitive varieties gets mentioned. And I have annoyed myself over the years giving integrated weed management talks where I suggest competitive varieties because without fail, I get the question, well, where can I find out information on if my variety is competitive or not? And unfortunately, that isn't information that's available to producers. And so the question became, is there a way that we could develop a strategy or a protocol where we could start ranking our crop varieties for competitiveness? Now, again, I know it's not going to be the top reason you choose a variety, but the idea is hopefully if you were down to 2 varieties that have similar yield, similar maturity, similar disease packages, but you got a resistance problem, maybe you'll pick that more competitive variety of the 2 to help give your crop that advantage over the weeds. So we set up some experiments in both wheat and barley. Those are in separate experiments. And we're looking at, in particular, the ability of those varieties to compete, which means that they reduce the weed biomass that is in there, as well as their ability to withstand competition, which means even with weeds in there, they're still yielding really well. So those are sort of the 2 factors that we're looking at. We incorporated different classes. So we had CWRS versus CNHR. In barley, we had malt versus feed, and those types of things, trying to see if there was a class link. And the biggest question was, can we rank these varieties for competitiveness? With a secondary question of, in order to rank those varieties, do we need a weed community or can we just use a single model weed? The reason behind that is, well, folks like Charles, Shaun, and I are perfectly happy to seed weeds into our research trials and establish them to make sure that we're having an effect. Most people don't really want to seed weeds in their research areas or in their fields. And so the goal was to see if we could do it with just a single weed or a pseudo weed instead of actually having to establish that weed community. So we did this at 4 sites, Lethbridge and Lacombe in Alberta. And then we did 2 sites in Saskatoon, one with Shaun at Ag Canada and one with Chris Willenberg at the University of Saskatchewan. Now, the difference between the 2 Saskatoon sites is that Shaun used a weed community and Chris used a single pseudo weed. So they're not at identical locations, but they're pretty close to each other. They would have experienced fairly similar weather conditions, and that major difference would be between sort of the weed component of that study. So the way this is set up, you're looking at 2 replicates in one of the trials here. The varieties are running left to right across your screen. Okay, so they go through this way. Then we have a weed-free and a weedy part of each one of those replications. Okay, so on the left-hand side, the back part of that rep is weedy, as you can see from the very heavy green pressure. And on the right side of the screen, the front side of that replicate is our weedy area. In wheat, we looked at 16 varieties. You can see primarily CWRS varieties, some CPS, some CNHR, some CWSWS, and CWHW. The way we broke up the number of varieties in each of these classes was based on insured crop insurance acres, which may not be exactly accurate, but gives us some representation of what's being grown out there. I'm not going to spend a lot of time talking about the specific varieties. I wasn't as concerned about giving each one of these a hard and fast ranking in this. I wanted to know, could we rank them? In barley, we had, I believe it's 13 varieties. So we had some malt, some 6-row feed and forage varieties, and then some 2-row feed. We took a lot of data, of course, as we always do, looking at emergence dates and counts, but we also wanted to look at traits that might be linked to some of the competitiveness. So things like, how fast does it cover the ground? What's the leaf width? How much biomass does it produce? How quickly does it close the canopy? How tall does it get? Does it lodge or not? What's the maturity? What's the yield? And then doing some quality factors like 1,000 kernel weight and dockage. To get into the results, I want to start by actually stepping back to that crop type. So these trials were not set up to look at crop type. They were pulled apart with wheat and barley separate. However, those trials were side by side, okay? They were seeded within a day of each other. We seeded the same seeding rate of weeds at the same time, at the same depth. Everything was the same for the most part, except for that crop type. Even our seeding rate was the same. This is our barley study here on the left. So you can tell which is the weedy and weed-free components of these trials. But relatively clean. Yes, there's some weeds there, but it's not awful. This is our wheat. You'll notice the weedy side of the trial looks a little bit like a little green forest down there. Okay, now I get pushback saying, well, you've got your pathways sprayed out on one side and not on the other side, and that's leading to why it looks so much worse. So I blacked it out. So what you can see now is just the replicates, and there's just quite frankly a heck of a lot more weeds in the wheat trial. Okay, again, a day difference between seeding, same seeding rate of the same weeds. Okay, crop type is still a valid recommendation. If you've got a massive weed outbreak, you might want to consider switching up your crop type in your rotation to help you manage that. Not going to say any more than that. Could we pull these varieties apart and rank them for competitiveness? Yes, we could. Okay, so on our bottom axis here is our ability to withstand competition. That's the ability to yield even when weeds are present. The higher it is, the closer your weedy yield is to your weed free. Okay. On the other axis, on our vertical axis, is our weed biomass. Now I have flipped this, so there's lower weed biomass at the top and higher weed biomass at the bottom. Okay, it just makes it easier to understand the different quadrants. We put the average weed biomass in here, and we put the average ability to withstand competition in here. Those are the dotted lines. So if you are better than average in terms of yielding when there's weeds there, and you're less weed biomass than average, you're probably highly competitive. If you have a low ability to withstand that competition, so you have a low yield, you're really impacted in your yield when weeds are present, and you got a lot of weeds, now you're going to be in the low competitiveness category, and everything else kind of falls in between into an intermediate. Now, all those lines going every which way, that's showing variability. This is across all our locations and all our years. So there is quite a bit of variability that occurs year to year, which is why we wanted to keep it relative to one another where they've all experienced those same conditions year to year. Okay. So what I want to get across here, again, I don't want the focus to be solely on the varieties, but yes, we can pull these apart. Now, the different colors to the different varieties indicate what class they're in. There's really not a class effect happening here where you can see, ooh, this class is definitely more competitive than the other, okay? So that, again, this is in our barley. Just another look at this. This is the list of our barley and where they ranked across locations as well as at each of our individual locations. So the first thing I want to draw your attention to is that there are some varieties that are fairly consistent in terms of their ranking. So let's look at, say, AB Cadillac here, which was primarily low competitiveness, was intermediate in Lacombe and across locations, but fairly consistent in that sense. Compare that to something like CDC Austinson, which was intermediate across locations, highly competitive at Lacombe, low competitive at Lethbridge, and intermediate at Saskatoon and U of S. What do we actually think that's going to be? Well, it's probably going to depend on the region. Okay, there's probably some regionality, some weather type responses showing up in here that is changing that competitiveness by region, which means we're going to have to look at this by region to help inform our producers. The other thing I want to draw your attention to is the comparison between the Saskatoon and the U of S, where some of them are fairly consistent, like Austinson or Canmore, but then you have something like Sirish, which is highly competitive at the University of Saskatchewan, but low competitive at Saskatoon. What's the difference between these two? Again, Saskatoon had the weed community, while U of S had only a single pseudo weed. So it's likely that different weed species in there are changing that ability of that variety to compete or withstand competition based on which weeds it's trying to compete with. Which means to get a good true ranking, you're going to have a community of the representative weeds in that area. Okay. Similar story with wheat. Again, pulled them out here into our 4 quadrants, highly competitive, low competitive, and intermediate. Again, you don't see a major clustering of different classes showing up here. They're a little bit in each of the different quadrants. Again, a significant amount of variability across the varieties and across site years, indicating that we're going to need to do this over multiple years. We're going to need to keep this relative to one another. Okay. Looking at it across locations and stuff, again, you see that same type of pattern where something like Brandon was maybe fairly consistent, something like Viewfield was maybe fairly consistent, but then you have something like Foray where you had a little bit of everywhere from low to high and everything in between. Okay, same type of story here. We're going to need to do this regionally. We're going to need to do this with representative weeds in each of those areas. Again, with that second point, looking at differences between Saskatoon and the U of S. So that's kind of the story that's coming out of this for me, is if we want to set something up to do this for producers, to actually give you these rankings, it's going to have to be regional, and we're going to need to use representative weeds. Did we look at those traits that I talked about and see if can we pinpoint traits that are affiliated with competitiveness? We did look at correlations. There are correlations that occur between some of those measured variables and either the ability to withstand competition or the ability to reduce weed biomass, but they're not overly strong from the preliminary work that we've done with them so far, and they're not always in the direction we anticipated. So I need to sit down and spend a little bit more time with those correlations and see if we can actually pick out traits that maybe we can feed back to our breeding groups and say, hey, if we can start breeding for this, we might be able to move the needle on competitiveness of these varieties. So that's the intention with the correlations. Still some work to do there in terms of the traits. So sort of our conclusions from this study is varieties do have different competitiveness. They can be broken into categories, but we need to understand the regionality, or at least be incorporating that, and how we work with it. And we would need to use weed communities. However, the question that I've started asking coming out of this, to screen all the new varieties in the registration process will take a significant investment in terms of finding locations to do this, setting up a protocol, setting up a group that is working on this every year. That needs to be regional to serve the producers in the different regions. And so my question is the efficacy of using a more competitive variety high enough to warrant that type of investment? Or does it not matter so much where even if it's a low competitive variety, we could increase the seeding rate and have as much efficacy? So we need to look at that relative effect of some of these strategies is sort of the next step for me for this, before we actually go and make this infrastructure investment, to make sure that it's actually going to be worth that type of investment for producers. So I'm going to take a little bit of a step back from some of the new management stuff and go into a little bit of a sticky diversion and talk about cleavers and a little bit of resistance screening that I've been trying to contribute towards sort of an addition to some of what Charles does. So I want to talk about quinclorac resistance in cleavers. Okay, so quinclorac resistance was found in the 90s. We've actually had this around for quite a while. The first time it was found, the field hadn't had quinclorac sprayed on it before. It hadn't actually had selection with quinclorac previously. It had been selected with other auxin herbicides. Now, cleavers are selfing, and the work that was done suggested that this was a recessive gene, which meant it wasn't going to spread very quickly. But we haven't really been monitoring for this biotype across the prairies. My concern came when we had MRLs approved in canola for us to start using quinclorac in canola. And in particular, in glufosinate tolerant canola, there's a tendency to use that glufosinate tolerance, but then also just add quinclorac to help clean up the cleavers because they can be a little tricky in glufosinate canola. And so we're seeing increased use of this product to manage cleavers in our canola systems. And so my question was, what's going on with that quinclorac resistant biotype in terms of how common it is? So once Charles was done with the cleavers from the prairie herbicide resistant weed surveys, we took what was left of those seeds and started screening them for quinclorac resistance. So for the 2014 to 2017 survey, there was 31 populations from Alberta that we screened, and we had 11 that showed some survivors of about a 4x rate of the field rate of quinclorac. Okay, so we are 4 times above the field rate, and we had 11 populations with some survivors. 2019 to 2023, we had no populations with survivors from Manitoba or Saskatchewan, but we had 6 populations with survivors from Alberta. So on the right-hand side is an example of some of those survivors. So you can see they're not just gasping for air and hanging on, they're pretty healthy and happy in there. Okay, so that was a concern. So with the 2019 to 2023 populations that had survivors, we grew those through to maturity and we harvested the seed. And we picked a couple of representative populations to do a full dose response study on, or a resistance screening assay like what Charles talked about. So we grew our cleavers in single cell trays, we sprayed it in a moving track spray chamber, and we went all the way from untreated up to 27x the field rate. So 27 times the field rate of what's being used in producer fields. This is still preliminary data, but this is what this dose response looks like. So our susceptible population is on the bottom, and then 3 populations that we were concerned about above that. Now, population 2 and 3 did not have great germination. As you can see, there's not a lot of plants in there, but the survival still is telling a story, I think. The rate in the middle, that is 101 with the green box around it, that's our field rate. Okay, so we start on the left is our untreated, and we go up to all the way up to 27x on the right-hand side. Just looking even at population 1, that's an awful lot of green plants at 27 times the untreated dose. Okay, so all of these populations have survivors at that 27x for those populations that we were concerned about. This is just that 27x rate. You can see the left-hand side, they're very toasted and dead, and you can see very green, happy plants in the other 3. On the far right one, you'll notice at the top, sorry, just going to jump back, on the top right corner of this tray, that's actually a new cotyledon that emerged, and quinclorac does have some residual, and we still had a new plant pop out of there. So I obviously haven't done the stats or anything yet. Looking at this visually, I'm going to say we did find additional quinclorac resistant populations. They do seem to be centered in Alberta for right now, but they may be a little bit more widely spread than we thought they were, although it's still not a massive proportion of the population. So again, more work to do on this. It is preliminary, but some interesting results there and some very happy cleavers that I had really hoped would not be so happy. Along with that, we're just finishing up the second round of this in the greenhouse, sorry. So we are pretty sure we found some additional resistant populations. We need to map them out and see if they're centered on a geographic region or if they are spread across the province. And we need to go back to some of those initial screenings of all 6 populations and the 11 from 2014 to 2017, again looking at that regionality, because based on what we've seen, if we mark them as sort of survivors in that single dose screening, it's a high likelihood that we did find resistant biotypes. In addition to that, we're also doing some screening with glyphosate. Now this is super preliminary, but I just want to bring some awareness to what we're doing here. We did a single dose screening just like we did with the quinclorac, and there was enough regrowth to prompt us to screen a couple of populations in a dose response here as well. This is currently ongoing. We just seeded our first round of this actually yesterday. When I look back at our single dose screening, we had 20 populations with regrowth through our glyphosate single dose rate. 5 were particularly concerning with their ability to grow through. So we are digging into this further. We're trying to see what's actually happening here Glyphosate screening can be tough. I do want to highlight that. Glyphosate is a product that when you're doing controlled greenhouse or dose response screening, plants don't like to die as well to it as they do in the field for whatever reason. Plants can be a little more stubborn. Glyphosate can be a little more stubborn. You don't get as good of a kill in a control condition. And so you get variability in these greenhouse conditions. But I do think it's worth a second look with particularly these 5 populations where we were seeming to having consistent regrowth through that single dose screening. So trying to figure out what's going on there. I'm a little bit concerned because glyphosate and quinclorac tolerant cleavers would be a problem for canola production in particular, as that's sort of our 2 tools for managing cleavers in canola production. I don't want to cause a big scare with this, but I just want people to be aware that, you know, we are looking at this and there is some concern around this situation, I guess. We will need 2 rounds of screening. So like I said, we're just starting our first one. We just seeded it yesterday, and we'll be digging into this a little bit more in the next couple months. The cleaver screening is funded through CARP, the Canola Agronomic Research Program. And we're trying to figure out what's going on with these particular plants. A couple key takeaways from the last little bit. Harvest weed seed control does show promise. The mills show promise, and we are seeing adoption. Again, there are some challenges around green material, but there are ways to manage that. Directed energy is a new harvest weed seed control idea that we're hoping will be possible for lower cost and in particular older and smaller equipment. And some of the very early indications are that it definitely does have an effect on seeds, but how that all works out on all the different species will be really interesting to see. Competitive varieties, we can do it, but there is that question of how does it compare to other strategies and would it be worth that infrastructure investment? With cleavers, we're trying to catch some problems early on before we're having a lot of complaints from producers, and we're trying to track these older problems that may start causing management issues in our current situations. With that, if you want to ask me any questions on anything and you don't want to ask them today, my contact info is there. I'm existing on pretty much every social media site there is. My email address is there, and if there are questions, I'm happy to chat about them. Thanks.
Great. Thanks so much, Breanne. That was fantastic. The first question we have is when you were talking about the directed energy, does that give off any heat at all within the combine?
It does give off heat. We are still sort of looking at that level of infrared wavelength light, which is what gets converted to heat, to determine how low can we keep that to make sure we're not starting a fire risk. The indications from people that I've talked to previously and sort of on that commercialization side is that it will still be a lower fire risk than all the moving parts that are incorporated with the mills. I don't know what data they're getting that from, but there is some heat generated, and so that's definitely a big question on how low can we keep that heat level to make sure that we're not inching towards more fire risk on our combines, because that's bad enough as it is.
For sure. Thank you. The next question is, when the barley and wheat varieties were ranked, the weed biomass scale was larger for barley than wheat, despite the visual appearance of the barley plots having lower weed biomass. Do you have any thoughts on that?
Yeah, so that picture, that image that I showed was from the Lacombe site in 1 year. Between years, we had a lot of variation in terms of how well our weeds established, just like with our producers. When it doesn't rain, things don't like to grow when we're trying to grow them. And so it was averaged across all of our site years and all of our locations in those particular graphs. So I can go back and I could tell you exactly which location had the higher biomass in the barley that caused that to bump up compared to the wheat across locations, but it was essentially a single site year that kind of changed that scale a little bit on us in those averages. So the visual was from a single site year in Lacombe. It just was such a striking visual that I like to show that comparison. And again, we didn't take any data on that. It was just the visual from our site in Lacombe. But those axes are related to being averaged across sites and averaged across years.
Perfect. The next question is, when you had winter wheat and peas in rotation, what percentage of efficacy on wild oat production did you see?
In terms of percent control, I'm assuming. I don't know that I actually have a percent data. We were looking at our number per square meter was our biggest sort of marker that we were looking at, and I didn't look at a percent reduction compared to the other rotations. It was just a notable reduction in our density and in our biomass. And a statistically significant one, but I don't actually have the percentage. I could go back and calculate that. I just can't do it off the top of my head while I'm attempting to answer.
Fair, yeah. That's the last question actually for right now. So thanks again for presenting. Yeah, that was fantastic.
Thanks.
Our next speaker is Dr. Shaun Sharpe. Dr. Shaun Sharpe is a research scientist with Agriculture and Agri-Food Canada at the Saskatoon Research and Development Centre in Saskatoon. Dr. Sharpe works primarily on high-priority herbicide resistance weeds in annual agronomic systems such as kochia and wild oat. Dr. Sharpe's research involves herbicide resistance prevention and mitigation, integrated pest management, and developing alternative control techniques. Dr. Sharpe received his bachelor's in science from Mount Allison University in Sackville, New Brunswick, his master's from Dalhousie in Nova Scotia, and his doctorate from the University of Florida in Gainesville. Dr. Sharpe joined the Saskatoon Research and Development Centre in Saskatoon in 2019. Dr. Sharpe has authored or co-authored over 60 peer-reviewed research or extension articles and currently serves as a governance director for the Canadian Weed Science Society. We can start whenever you are ready, Shaun.
Thank you. And are my slides coming through okay?
Yes, yeah, they look perfect.
All right, perfect. Thank you. And good morning, everybody. So today I want to talk about two project streams. One is going to be on kochia and how to manage it on the landscape. And the second one is going to be work that we are conducting to try to keep the problematic pigweeds out of the prairies. We'll get right into it here. So this first study is going to be from some research that we started between 2021 and 2023 that was funded by ADF through the province and in Sask-Wheat. And this was evaluating physical control strategies to manage kochia patches and the effect on the soil. With the time limits today and the amount of material I want to present to you, I'm just going to focus on the kochia data, but that QR code will take you to the manuscript. So all of this will be on there as well. So for this work, we were interested in doing patch management with kochia. Now, kochia is a tumbleweed, and it is patch forming. So where those tumbleweeds deposit, typically there would be a high deposit of seed into the soil seed bank, and then it forms these dense monocultures. One of the chinks in the armor, or the kinks in the armor, sorry, for kochia is that it does not last long in the soil. With previous research, it lasts up to about 2 to 3 years, with most of it dying after the first year. So if we are to intensively manage the areas where the kochia is existing, we can hopefully get those populations down. So for this research, we looked at 6 treatments. The first was a non-treated control where we permitted the kochia to grow. Then we used a mowing treatment. For this treatment, we went out about once a month when we were doing the counting data, and we would count and then mow. We did attempt to establish AC saltlander, which is a green wheatgrass, but unfortunately we weren't able to get it to establish, so I'm not going to present on that. We also used black plastic mulch, hydro mulch. For this we used a coconut-based mulch, and then we used chaff from the field. This was largely derived from the previous crop or the environment around it, so we used a lot of wheat chaff, for example. At 1 or 2 sites we had to use kochia biomass, and then at one site where the producer had tilled, prior to us putting the trial out, we did use some cattails from a slough, although I'm not going to advocate that practice. We were just using biomass from the area to try to bury the kochia. First, I'm going to show you the mulch depth. So this was a 3-year study. So We started in 2021 and we kept the plots up for the next 2 growing seasons to the end of 2023. This was largely on the margins, so we did not grow crops through the mulch, so it was just the kochia and then the treatments. So I've used a bar here, just put it on the laser pointer, and so this was about the average depth in 2021, and then the black bars is the chaff treatment, while the gray bars are the coconut mulch treatment. And as you can see, there was quite a difference between the two treatments in 2021, but by the time we reached the end of the growing season in 2023, the chaff layer had reduced either from compression or from the wind in the environment. So next, on the next couple slides, I'm going to show you the kochia densities in each of the treatments. I've only got 4 of them on the screen, as when we used the black plastic mulch, there was no kochia growing through it. So while that's a, a good result, it's one that's hard to graph. So I have left that out, but I will show it to you at the end. So we have kochia density in the y-axis, and then our timing is on the x-axis with each of the treatments clustered in the timing. So it goes untreated, cocoa mulch, chaff, and then mowing. And as I mentioned before, we had the students counting before they would mow. So that's why we see at the first measurement timing in 21, we do see the higher bar, and then we do see it go down over time by the end of August, or the middle of August, we do see those populations diminishing. When we did nothing, in this year, we had about 400 to 500 plants per square meter. This was one of the best years for kochia suppression. When we moved into 2022, and again, these are the same plots from 2021, we start to see that the kochia density is getting out of control. We're getting up to 10,000 plants per square meter. And if you look across the measurement timings, we do see a bit of a curve, a bell curve. I think there was some natural competition between the kochia where some of them did die out. And then when we used the chaff treatment here, this was able to keep the kochia at the lowest density, and it was about the same as the previous year when we did nothing. So there was still quite a bit of kochia there, but they did get quite low by the end of the growing season. This year and the previous year were very droughty, so there was not a lot of precipitation in the environment, which did favor kochia. So one of the uncontrolled variables in the study was the amount of kochia that was tumbling through the study. So I think there was actually a lot of seed deposited in that way, since it was kind of in areas where kochia was already, so there was a higher risk of invasion. So I think part of that is what we're seeing here as well as just the drought and kochia being quite productive. When we move ahead to 2023, we still see in the untreated plots, we're still getting up to about 2,000 plants per square meter. That's a lot of kochia to try to manage with any treatment, particularly a herbicide, which is why we want to try to use some of these techniques. By the first measurement timing, all of the other treatments were about the same, but with the mowing treatment, we do see good success by about early July, and these populations are going down. Now, we didn't really see that response in 2022, which I'm going to show in this next one with the mowing, and then we did get back to very high efficacy in 2023. The kochia in 2022 was very, very short. It was not over my ankle, whereas in 21 and 23, it was over my knee and higher. We haven't quite been able to figure out why the kochia is doing that height difference between the years, if it's largely environmental or density, although we have done some work in the greenhouse and it doesn't appear to be density related. So Here again is the black plastic mulch, and it is at 100% control. We did have to go out and patch up a bit of that black plastic mulch due to degradation in the sun. Then we have the chaff treatment and the mowing treatment, which performed quite well. And then the coconut mulch treatment performed well in the first year, but we started to see its efficacy decline. Sorry. So this next slide is the economic analysis that I conducted on this study. We used the inputs from the study to estimate the cost of trying to control a kochia patch that was derived from Charles's previous work from 2019 and 2020 for the HR survey in Saskatchewan. So from that, we were able to estimate how much of the field was infested with kochia, and then how much it would cost to try to implement these treatments at that scale. So we have a category here for equipment, one for materials. So equipment, we have 5 options here. So for the chaff and straw treatment, I used a cart, which is why there's some equipment costs here, or for bales, which is derived here in the materials. And then the labor is about the same to apply them. Then in the coconut mulch, there is a cost for an applicator as well as the cost of the mulch. And then here we have quite high costs for gas and labor, because with the coconut mulch, it would require a water source. So you actually had to run 2 tractor trailer or two tractors, to pull that out. With the black plastic mulch, there's no way to do this commercially with a tractor. There is plasticulture equipment, but it's not really for this setting. So I did this estimation via hand. So that's why there's a higher cost of labor, and it's largely the cost of the black plastic mulch. And then with the mowing, cost for a pull behind mower, and then the cost of going out 4 trips per year for 3 years, which is the big difference here compared to all of these other treatments, where we did estimate for the black plastic mulch some time for labor to go back out and repair, but otherwise we did things once and then had an effect for 3 years. So with this, we found that the chaff cart, the chaff with the bale and the mowing were the most cost effective, but with the mowing, it would be a limit of growing the crops there because it's hard to be selective with mowing. And then to follow up on this with the straw, we are doing some work to evaluate if we can grow cereals through straw, which it turns out that we can, of course, although there is some lodging at the higher rates that we're using. For this next study, it is one of the sites for the Bridge to Land Water Sky, which is in Indigenous Living Lab that I participate in the province. They're in Treaty 6 territory, so north of Saskatoon here. And we're working with Mistawasis in Muskeg Lake Cree Nation, and we are evaluating BMPs at field scale. And so for this work, this is kind of a biological and cultural approach to managing kochia, where they are doing marginal land conversion. So if you've heard some of my talks on BMPs, this is one of them where we want to have good weed management on the borders. So in this effect, they're evaluating where the borders are and then doing marginal land conversion to actually put them into borders instead of trying to grow a crop there. In this particular site, there was an infestation of kochia that went back a number of years, and it was largely due to the salinity of the site that they thought it was being established, although there are other reasons to do marginal land conversion, including topography and poor drainage and other characteristics of the soil. So this is actually from the site for the establishment of the forage in the spring of the year after. This is just another picture while we were out collecting counts. So with this forage, it was seeded in early May of 24, and it was a blend of salt-tolerant grasses and alfalfa, and they did include a bit of the Ducks Unlimited Pollinator blend as well. And then with my team, we went out to measure kochia at 2 timings, early in the spring, which is kind of this timing here, and then right before they would harvest. So this is just a picture of the kochia on the site in the spring, so very close up. This is the button stage or the carpet stage kochia, if you will. So it's still very small and forming rosettes, and this is a dense carpet that comes up. One of the main results that we found in this is that in the forage, we were not seeing the emergence of these dense carpets in the spring compared to in the cropland. There were still plants there, of course, because there was kochia in the area historically, and with the tumbling dispersal, there is going to be seed rain across the site, so we are still seeing kochia emerge in the forage, but because the forage is emerged early and able to use the space and the light and the resources, we do see less kochia emerging and establishing there compared to the cropland. And this is across the 2 measurement timings. I did have to use a rank transformation here because of the high variability in the kochia densities, but it was consistent across the 2 timings. So there was always a lot of kochia in the cropland and a lot less in the forage across the 2 timings. All right, so next I'm going to pivot and I'm going to be talking about invasive pigweeds. For this presentation, I'm going to focus primarily on Palmer amaranth. But a lot of these considerations will also extend to water hemp. On the screen now is a manuscript that I published, I think in 23, to kind of bring awareness to this problem. And I'm going to talk through a bit of my reasoning about this. The QR code will take you to the DOI link for the manuscript, where there is more information about this there. So Charles very kindly introduced you to the biovigilance spectrum. I just want to reintroduce it briefly here because for this project, since we largely don't have Palmer amaranth in the prairies, there's been 1 detection so far, we will be working up here in the awareness stream and then in the understanding step as well. So bringing in awareness of how these pigweeds can be a threat and then understanding the ways that they can invade so that we can try to stop them, as well as some information about how to identify them as well. So here's just a picture from the Ontario government to show the characteristic seed head of Palmer amaranth. So I just want to talk a little bit about the origin stories of Palmer amaranth. When I was going to grad school in the States, I know there were colleagues of mine there who had a different opinion of the origins of Palmer amaranth, where they found it in the Saron Desert in the 1900s, and they think that was the starting point. I tend to take the word of Sawyer, where he had reported that Palmer amaranth in particular was used by indigenous communities to the Saron Desert region, which I'll show you on a map here, where they used it as a pot herb and a grain, and that they would sow it out with their maize or their corn. And if the corn grew, that was great. They could use it for food. If the Palmer amaranth grew, that was great. They could use it for food and in their ceremonies. And again, they're using it as a grain. So kind of this part here would be harvested and made into breads and other things, whereas in a pot herb, they're going to be harvesting very, very young plants, and then they're going to eat the leaves by boiling them in water. So in 1955, Sawyer did explain that he didn't see any evidence of domestication of Palmer amaranth, but I would suggest that this may have been hidden because of colonization, particularly of the Spanish in this area, which was quite disruptive of the communities that were there previously. So we may not have that information. But if we look at comparative plant anatomy and botany, we do see that the grain amaranths that we do know were used for grain have a very similar seed size to Palmer amaranth, whereas if you look at water hemp and other pigweeds that are more weedy, their seeds are typically smaller. In Palmer amaranth and water hemp, there are no hairs on the stem or the leaves, which is a trade-off that's seen in kale, for example, where there's a taste and defense trade-off. So those hairs would be used for defense, but we are putting an artificial selection on that so that those hairs are not there anymore, because we are picking the ones and growing the ones with no hairs because of taste. And because of the dual use of being both a pot herb and a grain, this could also make it quite difficult to tell, because now we have two selection pressures for different parts of its life cycle. This is an image from farmers and their language. The first expansions out of Science in 2003, so it's a bit of an older paper, but what they do is they show you all of the epicenters of crop domestication. Typically, I've shown this for talking about the Fertile Crescent, but for here, I want to show you this area right here is where they found Palmer amaranth historically, which corresponds to the areas where there was thought to be agricultural spread through language and carbon dating of artifacts. So if we move ahead to the 1950s, so again, this is kind of the area originally where they were farming. And then we see Palmer amaranth has actually spread down into Mexico and then eastwardly towards Texas, right about here, and is starting to overlap the range of multiple other pigweeds. One note that this particular botanist had made is that on the herbarium records for Palmer amaranth, it was found at this point, this is well before the use of a lot of broadacre herbicides, it was already found about half the time in artificial habitats and half the time in natural habitats. And those types of artificial habitats could include irrigation lines and roadsides, places where humans are trying to manage the landscape. So these are some custom maps I did to help show the spread of herbicide resistance in Palmer amaranth. It was first detected in 1989. And so this is the first 10 years of observations. Here is where I'm at in Saskatchewan as a point of reference. So here's largely Kansas and Arkansas are going to be the two hotspots to forecast a little bit. And then we can see here in the Carolinas and Tennessee. But if you jump ahead to the last time I updated this map, which was 2023, red would indicate two modes of action, and then the brighter it gets, the more it is after that. I think these are 5 to 6 way modes of action groupings or stackings of different herbicide resistance traits. And as you can see, it's spread all up the East Coast and then through the plains as well. There's no tag in Minnesota. They have definitely found it there, but they have been working on eradicating it. And as far as I'm aware, they have been successful thus far. Whereas very close to us, and even I think in Montana now, they are detecting it. So it is moving northward to the border. This is not necessarily that the populations in each province reach the levels that we are seeing any novel mutations or adaptations. It's likely more a combination of a high population plus the spread, and the spread is bringing those traits into new areas as well. So here are just some pictures to show you the two seed heads of them, just in case you happen to see them out as you're scouting. This is Pomerantz here, and this is water hemp. So again, one of the telltale identifications that they used in Minnesota for their eradication work was that the seed head kind of was very characteristic. It was easy to tell that that was it. And I would argue again that it kind of looks like a grain amaranth. I'll show you some pictures from the ones we were growing compared to this. And then this, of course, is because the flowers are a lot larger and that they do have these spiny bracts on the female flowers of Palmer amaranth. So for identification as well, two of the main traits are going to be a hairless stem, and that is for both Palmer amaranth and water hemp. So here's just a good example here. But this is not going to get you to the species level. Another way to do this is going to be through the petiole length. If you flip this leaf kind of at this section here, the petiole or the stalk of the leaf is typically longer than the leaf blade itself. So this won't reach this axis if you flip it over. Now this is a little bit tricky because it's going to manifest largely on the older plants, on the older growth. It may not be present on the younger growth. There's also been observations that Palmer amaranth may have like a print here, but it's not characteristic across all the plants, so it's not very diagnostic. So now I'm going to get into a little bit about why I care about this weed and why I don't want it in Saskatchewan or the prairies in general. It's because it's very invasive and it's a huge yield robber. So in corn, for example, in Kansas, which I was mentioning is the hot spot for a lot of the HR, they're seeing up to 91% yield loss in corn, and corn is quite competitive and has a good profile for herbicide tolerance, much like our canola. Whereas in corn, they're seeing nearly 80% loss to Palmer amaranth, nearly 60% loss to water hemp. And again, this is in Kansas, so this is in the Great Plains. And here's another example of why I care. Again, this is in the manuscript as well. For Palmer amaranth, these are all of the different groups that it has developed HR to. That's not to say that every Palmer amaranth is going to have all of these different groups. It's 2, 3, 4, 5, 9, 10, 14, 15, and 27. So the 10, the 3, and the 27 in particular are modes of action that we are using to manage kochia in different systems. So this is a weed that's already slipped through some of those. Some of the big ways that it's doing that is through metabolism, particularly this group 3. They found a population in Arkansas, I believe it was, that was able to metabolize the group 3 herbicides. There's also target site mutations and altered absorption, translocation, and gene amplification depending on the type of resistance. So there's multiple types of mechanisms that it already has adapted towards. And then with the situation with water hemp, it's the same, but a bit less. So there is no group 3 resistance. There is group 9 resistance and 14 though, but we don't see that group 10, which we're using in the canola, which is good. But again, we do see the metabolism is present for different modes of action. So we want to be careful. There's also target site mutations and gene amplification for the glyphosate. Here's just a picture to give you a visual. This is out of Tennessee in some extend soybeans. So these would be dicamba tolerant soybeans. And it looks like they're growing a good crop of amaranth, but that's not really what they're doing. And they really have a Palmer amaranth yield loss issue or a infestation issue. So this is all Palmer amaranth picking up here, or that's poking above the soybean. So I wanted to go over the different ways that it's able to spread because previous research has shown that the original invasion out of the Shaun Desert ecoregion was not necessarily due to environmental parameters alone. And kind of when we look at the literature, we see the large way that it's able to move around is by humans. We are the vector. And we are the vector in a number of ways. It could be through the growing of crops, it could be through the production of animals or the feed for them, it could be on equipment. So I have them listed here. This is just from clip art if you've seen these cattle before. So there's been examples of contamination of seed in plant propagation. So with millet seed in particular, I hear this one quite a bit because with millet, it's very, very small and black and shiny, and it looks a lot like pigweed. So trying to screen that out is nearly impossible. So if you are getting any millet seed from the States, be very, very careful, please. With native plant seed, this was a case of contamination in Minnesota, where they had then spread it over about 80 acres of, of native land, where they were trying to restore, so they had to go back and clean that out with fire and herbicides and other methods, hand pulling. For field contaminations, of course, corn, soy, and sunflower. This also includes the crops we grow here, since the palmer amaranth is reaching further north, so wheat, there are cases there. And so it can move through those systems in the grain. And then, of course, on equipment, there's been lots of cases of people noting, I bought a combine in the Carolinas and brought it northward, and now I spread the palmer amaranth that way. I've heard those sorts of stories as well. And reports on the animal husbandry side, there's reports of it moving in cattle feed. So when we take the screenings from a contaminated field and we feed them to the cattle for food, palmer amaranth persists in the screenings, they persist through the cattle, and then they can establish in the field that way. So through that could be in the manure, which can either move into the pasture or it can move outside the pasture, depending if that is going to be sold to growers or gardeners. So it also, as I had mentioned previously, it's been noted on railways and roadsides. That's another place that can be seen. Those are areas where we are managing, so they're areas where there could be disturbances. And then lastly, I did want to summarize the other ways it does spread. It is us, but it's not just us. So it does move in the wildlife. I'm going to talk about the ducks here in a minute. But this is harder to document since it's harder to track the movement of animals in your area. It also moves in water, particularly flowing water. So there's very, very small seeded, both palmer amaranth and water hemp. So if there's a good rain, it can flush those seeds down into a new area. We can move them into waterways. And then with wind, it does not necessarily have any adaptations to move in wind like we see with kochia or foxtail barley, but the seeds are very small seeded, or the seeds are very small, sorry. So if there's good wind, which we do have here in the province, it can help move that seed away from the parent plant, but largely its main mechanism is going to be us. We are the vector. There was some research to evaluate the movement of weeds in particular, with a focus on palmer amaranth and water hemp for this presentation, by a group in Missouri previously. So this is from a table where they worked with local hunters and they harvested ducks across 2 growing seasons or 2 field seasons. And then they looked at the contents of different parts of the ducks and then how much weeds would grow out of that. And so here, this is just to show you that they are feeding on, because these are ones that they hunted out in the wild, so they are feeding on both of these weeds and transporting them, okay? So they did do some feeding experiments. Since the results here are not super high with the palmer amaranth, I don't have enough time to show you all of them, but they did do some feeding experiments with mallards in particular, and it does pass all the way through and stay viable, although there is some degradation. So this was the big figure that got me from their work. So they looked at the rate of decay of the different seeds through the waterfowl, and then they looked at the flight speeds and potential travel times and things like that, and then they estimated how far the spread could be from where the study is occurring in Missouri. Then as you can see here, there is good projection for Palmer amaranth and water hemp all the way practically through the prairies, as well as a lot of our other parts of Canada. So that means we're already technically under seed rain from the movement of waterfowl, but again, they are not the primary vector, but it will complicate trying to keep it out, because if it does move in the environment, it's going to be harder to enact policy to try to combat it, since the policy can't control the animals in the wild. All right, so for this next part, we did put in for funding to try to work on some of these ideas about how to keep it out, to build in awareness campaign. I did want to do some niche mapping. It had already been done for Palmer amaranth and water hemp, but the resolution into Canada was not very high, so we couldn't really tell where the good areas were going to be that it could establish or that were compatible with where it's already growing. So that's what this next work is. So this was funded through the ministry, WGRF, with SAS Pulse, and SFSDC, which is the Saskatchewan Forestry Development Commission. So we use MaxEnt to do the niche mapping. So what is ecological niche mapping? This is when we're trying to match the species, in this case Palmer amaranth, to the environment where it's growing, so that we can help estimate other environments that are compatible with it. Now again, niche does not necessarily explain invasion. And as I went into detail here earlier, we are the vectors. So this is not necessarily going to tell me where it's going to go, but it may give an indication of, if it does go there, where it's going to stick, or where it's going to have an environment where it's going to be able to grow and reproduce. So for this approach, we're using MaxEnt, which is the maximum entropy approach, and we did use eval, which is a R package for MaxEnt, because it does allow some blocking. There's some previous research on sterile where they did this kind of blocking to help control for the native area versus the expanded range of its invasiveness, which we have applied here. We have done, for the observational data, we use GBIF, or the Global Biodiversity Information Facility. This is largely going to be herbarium specimens, and this is complemented with iNaturalist. I did have my team, my technician and students go through and vet the images to make sure they were Palmer amaranth, since it can be a little trickier to ID. And because in that information there wasn't a lot of the Canadian observations, we did include some of that data at the county level for those observations, just so we have some data in Canada to help with the modeling. For the environmental data, we use WorldClim, which is a fairly standard approach, and it has a whole list of different rasters of environmental parameters that can help to identify the niche, largely climate and topography, which is elevation. So these were the final rasters that we used to identify the niche of Palmer amaranth based on its current distribution. The contribution is an indication of the amount of gain through the training procedure. So this is how much it helped when we're training it. And then the importance is when we take it out, how much loss do we get? So overall, it's kind of, these two are the big two. So temperature seasonality, this is the annual variation in temperature across the entire year, and then the maximum temperature in the warmest months. So we do see temperature driving this quite a bit. And then down here, we do have a number of precipitation ones that all have smaller contribution or importance. And we did include elevation as well, just to keep that topography in as best we could. Yeah. So So this first map is the overall niche. So the darker bluish color is the high suitability range. The lighter blue is the moderate suitability range, and then the green color is the low suitability range. Thankfully, the blue is largely staying down here into the United States, but we do see some stretching up kind of in this area here of the invader range, which is more of a low suitability. I'm going to take you across the prairies here, right to left, so I'm going to go east to west. So here in Manitoba, this is southern Manitoba, this is the county where they did detect Palmer amaranth originally, but through the niche mapping, we're not really seeing a lot of compatible niches. Now, that's not to say it can't invade here, it already has. That's just to say that the area is not compatible with where it's already at, but Palmer amaranth seems to work outside of this normally anyway. With Saskatchewan, sure I'm moving in the right direction here. So this is on the east side of Saskatchewan, so we do kind of see this touch of color here, just south of the border, south of Crotch, which is still over here. And then there's a little bit of color developing near Maple Creek in here. And then what I was a bit surprised about is actually the extension of the niche into southern Alberta. So there's actually three kind of areas here near Medicine Hat, kind of this area here, and then to the east as well. So I asked my technician just to go through and visually estimate the different areas. And these would just be towns and villages. There's nothing smaller than that. But we've got Alberta here, and there's a number of communities in Alberta and then in southwestern Saskatchewan. And we did include Carmen here because this was kind of the area where the Palmer amaranth was originally found. But thus far from our modeling, we're not able to estimate that as a compatible niche. But that's not to say it's not going to invade that way. So as we go forward, we are hoping to try to incorporate more human-mediated parameters such as land use or the use of equipment or irrigation, those sorts of things, to try to help incorporate different human-related parameters. So this next study is part of the same grant. And for this one, we did a field experiment where we wanted to look at cultural suppression of pigweeds. Now for this experiment, I did not want to seed Palmer amaranth or water hemp because I didn't want to be that person. So we used a grain amaranth as a proxy. So we got some grain amaranth from Ontario. It is publicly available. And then we used 3 different cultivars in this system, as I wasn't quite sure how they would all germ. It turns out they all germed very well. So this is just to give you an overview of the different crops. So we had barley, canola, lentil, peas, and wheat, and then we did seed them at 1x and 2x as standard rates of seeding for Saskatchewan. We had a bit of a drought into May, June here in Saskatoon, so our June heights, this is about mid-June, are a bit low on the canola and the peas, and then into July and August, things picked up. And then here's just our crop biomass to show you a representation of the amount of growth that the crops had. The peas obviously producing quite well. The canola was a little bit less. And then we actually had quite a bit of biomass in the lentils. So the next one, we did include a control where we did not have any crop. And this was just to see what would happen when there was no cultural suppression of the grain amaranth. We seeded at 50 seeds per square meter for 2025. So here's the treatment list here. So we have wheat at the 1 and 2x rate, barley, peas, canola, and lentil. And then the control at the bottom is the grain amaranth. So in August, so the grain amaranths were later to emerge. I think that was partly due to the amount of precipitation we were receiving, as well as their natural biology with water hemp and palmer amaranth. They are later emerging weeds as well, so it's not unexpected. So they really started putting on biomass into August. So I'm just going to focus on the August measurements. You can see here we do get some escapes in peas that are comparable to the control where we did not have anything. And then in the wheat at the 1x rate, and then in the lentils. And then this is a little more clear in the biomass. So we got over 209 kilograms per hectare of biomass out of the grain amaranth, but we do see good suppression in most of our crops. Peas was a little bit of a miss. And then in the lentils as well. And again, this is not unexpected, as my colleagues have mentioned, the pulses are typically a little bit poor competitors, which is why we do this kind of work to kind of see if these things help. So when we increase the seeding rate, we do see a trade-off and we do get better weed suppression. Just a few field shots here. This is one of the best examples we had to just kind of give you a visual of what it looks like when these pigweeds start to come out top and over top of the canopies. So this is in peas. This is one of the darker varieties that we had. There's one that was almost purpley on the seed head. Just a few more examples. This was in lentil. This was actually on the edge of a plot. I think it was between two of our grains. This is just an example of the different coloration from using the three different biotypes, or sorry, the three different cultivars of the grain amaranths, which made it actually a bit more challenging to go find them in the field because now we're looking for three colors. So for the last part of this project, we do have funds available to do genetic testing to confirm if you have palmer amaranth or water hemp. So this spring, we should have the contract in place with the service lab. So if you do have any problematic pigweeds and you're worried about them, my email is on the screen. You're more than welcome to email me, and then we can discuss. And then if it is suitable, you can donate them to my program. We'll have a look and see if we do think it's palmer amaranth, water hemp, or one of the pigweeds we have, like red root or green. And if we're not sure, or we think it might be, we'll send it along for genetic testing and then get back to you on that. So I'll pause there. This is just a picture of my team from last year. They do a lot of the counting and heavy lifting, so I'm very grateful for their help, and I will pause here if there's any questions.
Great. Thanks so much, Shaun. One of the first questions we have, and we actually get this a lot as crops extension specialists, when you had with a kochia to like bale and bury it, what about bale and burn it? Is burning a potential management strategy for kochia after you've baled it?
Yeah, that's a very good question on the kochia. I have been getting more questions on baling it and using it as a forage as well. So it's kind of the same vein of, can we destroy it after we take it out of the field? I would say yes, with the caveat that we are on the prairies. With the fire risk that it is, usually that material is quite woody. It is a tumbleweed, so just be careful if that's the approach you want to do. It will destroy the seed. I believe it should if you use it. So that should be effective, but we haven't actually done a lot of research with fire just because of the fire risk. So if you do try that, please let me know how you do.
Yeah, be careful.
Please be careful.
Yeah. How long are palmer amaranth seeds viable for? Is it like kochia, like a 2-year thing?
No, they're going to be a little longer. I actually don't have the number right in front of me, but I think it was more on the 5-year mark is about the median for them. So a bit longer. Yeah.
Perfect. And if you do find palmer amaranth in your field, who would you report that to, or what would be your first step?
Go to your province first, of course. So if you're here in Saskatchewan, it would be Corey Jacob. Although I would say go to agronomist first and then work your way up so that you have all of the support you need, and then eventually it'll also come to me. You are welcome to reach out, but I will defer you, of course, to my provincial colleagues since we're all working on the same team.
Perfect. I think that's it for questions right now. So thanks again, Shaun, for your great presentation. I'm going to pause right now and actually share the CCA code here once I can bring her up.